So yeah, my name is Helen Woolner, and this is going to be a bit difficult.
In the meantime, read it online.
Okay, so
Next slide.
So my mum is from a tiny little island called Moki, which is located in the Cook Islands, so the Cook Islands is an archipelago of 15 islands located in the South Pacific, approximately 3,000 kilometres northeast of New Zealand. She came to New Zealand when she was 16, and she met my dad here, many years later. Met my dad here many years later, who was a Kiwi, and settled in Porirua. So I've grown up in Porirua my entire life. I grew up in Cannons Creek and currently still reside there. I'm living in Waitangiroa. Okay. Do you have a clicker I can use?
No? Clicker won't work.
No?
Just
okay.
Just
okay. Yeah. So, like many kids, growing up, I had a number of things that I wanted to be, but my very earliest memory was that I wanted to be a scientist. And sorry, I wanted to be a paleontologist, but that changed throughout years. At one point, I wanted to be a teacher, and then a police officer, and then when I was introduced to computers, maybe something like a computer analyst, and then eventually I settled on wanting to be an artist in my later years at college. But then I ended up at a special school, He Huarahi Tamariki, where I was taught by this woman, Juliet Bellingham. So she's a former member of the Zonta Club of Mana, and she introduced me to science, and I thought I'd quite like to have a career in science. So I started kind of looking at what options were available, but at the time there was this popular TV show called CSI, and I thought, wow, forensic science would be great, quite a cool career choice to follow. So yeah, if you click. So that all pointed to me needing to go to university, which was a new concept for me. I'm the first of my immediate family to attend university and the second of my extended. So yeah, very new concept. But the other thing was that forensic science meant I needed to go to the Auckland University, and my circumstances at the time, I wasn't able to do that. So I instead did things a little bit of a roundabout way, and I thought I would go to Vic, so if you click, and do a Bachelor of Science, and then eventually end up at Auckland. But again, yep, things changed. And I was introduced to natural products. So my interest in the science of natural products was sparked when So first I should introduce what a natural product is. And so that's a chemical that is produced by an organism that is found in nature. So you and I are producing natural products at the moment. But my interest in the science of natural products was sparked when I learned about the swan plant, which produces toxins that make it distasteful to animals. But there is one exception. So the reason the plant does this is to defend itself from being predated or grazed on. But there is one exception, and that's the monarch butterfly. So, the monarch butterfly caterpillar. This caterpillar will eat the plant and is not affected by the toxins that the plant produces, but instead it stores these toxins away in its tissues, and they are then passed on to the monarch butterfly. So what that means is that the caterpillar itself and the monarch butterfly they've got these toxic compounds within them, which make them distasteful to things like birds. So if you click, if a bird eats them, they end up not feeling very well. But the bright colours and the patterns of the monarch butterfly and the caterpillar mean that the bird learns not to eat, and the fact that it got sick, the bird learns not to eat those insects again. So other examples of natural products that have been helpful to human in terms of their health are penicillin, which was isolated from this fungus, Penicillium notatum, back in 1929. It was a fortuitous discovery basically, so the scientist Alexander Fleming was looking at growing bacteria. He left the petri dish over the weekend, came back on the Monday. It was growing this mold, but around the area directly around that mold, there was no bacteria growing. And so he thought, whatever that mold is producing, the bacteria doesn't like. And that's how the discovery of penicillin came out. And then there's the other compound, Taxol, which was the other picture back there, which is an anti-cancer drug used to treat cancer of the breast and ovaries. So that's isolated from the Pacific yew tree. So you click. So yeah, natural products such as these two are special chemicals, that we call secondary metabolites, and it's these chemicals that natural product researchers are interested in. So even though you and I are producing natural products ourselves, we don't produce secondary metabolites. We don't need to. They're metabolically expensive to produce. So an organism that needs to defend itself from being eaten in order to survive is something that will produce them. So things like plants and insects, but not everything produces them. So my specialty was actually in marine natural products. And you might ask why? Well, our oceans cover more than 2/3 of the Earth's surface. And many of these organisms that live in the oceans are soft-bodied or slow-moving, and they lack the physical defense mechanisms. So things such as sponges, algae, sea cucumbers. And because they live in a volume of large water, they're very, very toxic, because as soon as they're released, they're rapidly diluted by the water. So what does this kind of research involve? Lots of diving, exploring the beautiful wildlife, scaring the wildlife, collecting organisms. So that's the most important part. And then we take these organisms back to the lab where we extract them. So they're extracted with a solvent, usually an alcohol-based solvent. And if you click, that extract contains a mixture of hundreds of different kinds of compounds. So what do we do with that? We want to get a pure compound, one compound, because we're interested in seeing whether or not this will have biological activity. So we place it onto a special medium that will separate this mixture of things. So on the left there is the extract sitting at the top in the red, and then as we pass solvent through, we can see a migration of the colours passing through that column. And the different colours would represent the separation of the different chemicals. And so as we continue doing this, we can see that there's quite a drastic change within that chemical profile, and that's how you purify the compounds. So sometimes that takes multiple steps. And then once you have a pure compound, you basically have to solve its structure. So we have some really cool machines that will give us the structural information, and we use these to generate fragments, and then eventually we can put these fragments together based on what the data is telling us. So then we form the full structure. So now I'm looking at traditional medicines, and the aim of my project is to discover the science behind traditional medicines used in Samoa, New Zealand, and the Cook Islands, using yeast chemical genetics. So chemical genetics is the use of small molecule compounds to perturb a biological system to explore the outcome. And why would you want to use yeast? So the yeast we use is Saccharomyces cerevisiae, which is basically just baker's yeast, the same yeast that you have in your kitchen. They're cheap. They have a doubling time of 90 minutes, which means it's really easy to grow them and study. And there are many of the genes that are present in this yeast that are homologous, so they have the same function as human genes. So we can use it as a model organism for human disease. And they can also be easily genetically modified to study chemical genetic interactions. So I've put that paper up there, fantastic yeasts and where to find them. because basically you can find them in our chemical genetics lab at Vic. Yep. So we have mutant strains of yeast, and basically each mutant strain has a single gene deleted from its genome, and we know what this gene is. And there are two libraries of mutant strains. So one that contains approximately 4,600 strains, where each strain has one gene deleted, a different gene deleted, and the other that contains approximately 1,500 strains. And together, these libraries constitute the entire genome of the yeast. Oh, yeah, sorry. So how do we use this to study traditional medicines? It's been used in our lab previously. So basically, we treat each strain with the traditional medicine, and mutant strains they're a mixture at the time, but mutant strains that are affected either by reduced growth or because they've died provide information about the molecular pathways that traditional medicine is targeting. And so we can use this to basically validate the use of the traditional medicine, but it also may provide new information about other things that the plant may be useful for in the treatment that is used traditionally, but is not actually currently being used for. So in support of this work, I was fortunately able to get the 2020's Onto Biennial Science Award, and I'm going to use that to travel to Professor Denise Tasdemir's lab, where I'll be using some of her facilities to look at the chemistry of the traditional extract. So at the moment here I'm doing the biology, but there she's got some technology that will be useful in helping me to determine what the actual compounds are within. So yeah, just final words of encouragement, because I thought there were going to be some younger children here. So yeah, it's not always easy to know what the answer is to when I grow up I want to be. But if you enjoy discovering new things, if you enjoy solving puzzles, and if you enjoy exploring your ideas, then you should give science a go. And of course, you can do it. So, yeah, that's it. And I'd just like to, I have many people to acknowledge. Yeah, it's been quite a long journey. So I've tried to put everything up there, and yeah, lots of the words have ended up quite small. Thank you.
Well, we'll just have time for like one or two questions. So is there anybody who feels bold enough to ask a question? Yes.
Sorry, was that me?
Yes.
That's really fascinating, and I wonder, in terms of your research in Samoa, are you looking at specific, have you already identified what you're looking at, and is it directed towards particular illnesses? Or how are you organising yourself in terms of all the possible?
Yeah, so because yeast are quite robust organisms, they have they have a system where they're able to actually pump out lots of drugs that other things wouldn't be able to. So we need something that's actually going to basically penetrate the yeast cell. So we're looking at anything that has been traditionally used as treatment for anti-microbial type infections, because if it's got antimicrobial properties, it might have more of a chance of penetrating the yeast cell. So I have actually collected from Samoa already, and that's what my target was plants that have been traditionally used to treat infections and stuff like that.
All right, one more question. If there is one. There was one. Yes, yes.
Hi, Helen. Great to hear you. I just was interested, when you first went to university, what was the toughest bit about making that transition? Having given up, wanting to be a policewoman or a teacher, but it sounds like a pretty tough journey. So
basically I started there were a few things. I started as an adult student, and of course, I think I wrote in my little bio that I had 2 young children at the time. So the toughest thing was basically juggling being a parent and a full-time student as well. I had a lot of family support. I couldn't have done it without them, and is the reason I'm actually still here, is because my mum is here and she was there for me. So it's my turn to give back.
Thank you. I'll ask you to, thanks.
I'm really sorry about the technical difficulty.
No need to be sorry. That was fantastic. Thank you so much. I learnt a lot. I know that one of the intentions of this Entree Award is to inspire girls into science. Well, I'm inspired, a little late maybe, but for those that you are talking to, I know they'll be inspired. Science is fun, and when you're a kid and you do science experiments and Michelle Dickinson like blows up things in her hands, you're like, wow! And then you go to like school and science is boring. And so to me, the inspiration of like, here's something fun, you were diving and doing something cool, and jigsaw puzzles of working it out, and it really helps people. I think that's immensely inspiring. So thank you, and here's a little gift for you.
Thank you. Thank you very much.